monolayer. This is some equipment that we will need to have successful cell culture. The first thing we have is incubator. The incubator is like a house for the cell, so it will create the ideal condition in terms of temperature, pH value, humidity, so the cells can grow. This is how you heard it is where we're going to manipulate cells, handle cells. For example, we can check the confluency, we can see some new medium in subculture to replace. We also need the water bath. The water bath is where we're going to warm up the calcium medium, PPS, nutrition, because for long-term storage, those reagents will be stored at 40 degrees Celsius. And before using, we need to warm them up to the right temperature, so which is 37 degrees Celsius. We also need the CO2 supply, because like I mentioned earlier, the CO2 and the sodium bicarbonate system is a very important pH buffering system that we use frequently. We have a vacuum system or sometimes a packaging system to remove the old medium or the waste for the use of PPS, etc. We also need some plastic plates, pipettes, and pipette A, etc. So those plasticware, most of the time, it means that we only use them one time. Incubator. So you can see, or you can have, most of the time, most of the lab, we have two incubators stacked up on top of each other like this. And for each incubator, you can have two doors. The first door is made from glass. You can see through it. We can observe the cell culture medium color. We can check the cleanliness of the cells inside environment of the chamber of the incubator. Outside is a little thicker door, and this, in the middle of this metal sheet, usually they have a layer of water, and this water is warmed to 70 degrees Celsius, and we also have water running around, which is this layer of metal here as well, to make sure that the temperature is always maintained at 27 degrees Celsius. And on top of this incubator, we have the electronic system to control CO2 and also the temperature as well. And importantly, at the bottom, on the bottom shelf, usually we're going to have a tray made up of glass, and it will contain water, because the purpose of this tray is to make sure that the humidity inside this incubator is always maintained at the ideal level. And this is the display of a typical lab for cell culture. So usually we have about 2 or 3 hoods sitting next to each other, and this is the CO2. Okay. So here we have centrifuge most of the time. They have some sharp containers to contain glass or some sharp like syringe or anything that can cut paper easily. So in here is the biohazard vaccine. You can see these, all of these parts from cells or any kind of pathogens. They need to be sterilized and also put here, and they will be incinerated, they will be burned to make sure that no pathogen can escape the environment. And this is another picture showing how we're going to work in the hood. So you can see here, this scientist is wearing a coat with long sleeves, and he's wearing gloves. And he's using pipette to transfer some solution to the flask. This is some flask that we normally use in the lab. And inside here is considered like sterile environment. So before and after the culture, we need to clean them up with 70% ethanol. And some new labs, they use some new hoods, and those hoods, they don't have UV lamps. But some of the previously made hoods, they still have a UV lamp. And these UV lamps are used to, for additional sterilization process because of the UV can cause cancer. So that's the reason why nowadays the manufacturer usually they remove the UV lamp on the hood. And this is another picture showing how we can arrange this incubator next to the hood, and this is the CO2 tank again. This is another sharp container. And for the pipette and dishes, we're going to have multiple sizes of pipettes depending on the needs of scientists. For example, this is a 50 ml plastic pipette. This is 25 ml. This is 5 ml and 10 ml, etc. And you notice that most of these packets, they are individually wrapped. So, and we only open this wrap inside the tissue culture hood only. And this is some different size of the plastic made tissue culture dishes. And we are going to use the size that we need that fit our experiment need. This is another kind of tissue culture class. We call them like, this one could be T150, T75, T25, etc. If you notice that at the mouth of each cap right here, we're going to have a filter. And this filter will allow air to go in and out, but they will block pathogens from entering this flask. Some of the flasks, they don't have this air filter, so we have to loosen the cap a little bit so that air can get in and out, so we can have a good balance of the pH system. Of pipetting, which is a very important tool that we use frequently in tissue culture. And the requirement for this pipette A is that it needs to be powerful enough for us to move solution, but without destroying cells. Because sometimes some of the pipette A may be too strong and that can break cellular membrane. So we need a system that is powerful enough, but also gentle enough for cells. So this is some of the key steps we're going to need to do in order to prepare a complete culture medium. So first we need a bottle of medium, and most of the time this bottle of medium just contains the medium and some solution, but we need to add on top of it. First thing is pseudomobile serum, and normally people add pseudomobile serum to a final concentration of about 10%. And some of the labs they can do 2.5% or 5%, and also sometimes even 0.1%, depending on the experiment condition. And pseudomobile serum, or in short SDS, is the most commonly used serum supplement for the in vitro culture of eukaryotic cells. And SDS has a very low level of antibody, high concentration of growth factor, which is very important for cell proliferation in vitro conditions. And so SDS, because of these characteristics, it can allow us to have the versatility and flexibility in growing different kind of cells. And we also need to add the L-glutamine, which is a very important amino acid, to a final concentration of 1% in most of the cases. So L-glutamine is not very stable. So at the moment of manufacturing, the manufacturer already adds L-glutamine in the culture medium already, but because of its unstability, L-glutamine will be degraded over time. So right before we grow the cells, or right before we prepare the complete medium bottle, we need to add additional amount of glutamine to make it up to 1% as a final concentration. And we can also add some additional antibiotics like streptomycin or penicillin or antimycotic, antifungal, to avoid some minor contamination by bacteria or by fungi. And after adding all of these reagents, sometimes we also need to add some selection marker like tubomycin or some indiscipline antibiotics like doxycycline or tetracycline, etc. But after we mix them all together, we need to mix them really well by shaking the bottle up and down several times. And for long-term storage, we're going to store the bottle at about 40 degrees Celsius, and we need to warm up the medium to 37 degrees Celsius before we use the cell culture. And this warming up procedure usually takes about 10 to 15 minutes, depending on the size of the water bath. And there are 4 phases of the cell growing process, and depending on our need, we're going to wait until sometimes decline phase or sometimes stationary phase. But most of the time, we only wait until the cell reaches exponential phase and we start the culturing cell. So the first phase of the cell growth is lag phase, which is right here. So the lag phase is after we feed the cells to a new plate, then the cells will spend some time and stabilizing, and they will start growing very slowly. And because they need to recover from the stress of the culturing process. The second phase is the exponential growth of the phase, and cells will divide after every 24 hours, most of the time, sometimes every 48 hours. And in this case, the nutrients and also growth factors are still abundantly available, so the cells keep growing as much as they can, as fast as they can. And in stationary phase, the proliferation will slow down and stop. And stationary phase is usually when, for example, like nutrients run out, or we don't have enough space for cells to grow anymore. So in that case, we need to pass them to another place. But most of the time, we try to switch the cells to a new place when they reach almost the end of the exponential phase. Because when the cells reach about stationary phase and the cell constituency is about 100%, meaning that they occupy all of the place in the tissue culture class, then we're going to have the growth complex inhibition, and sometimes that can induce senescence, meaning that the cells will become old. And that's something that we don't want, because when the cells become senescent, they will grow very slowly, and a lot of things change. And they're not normal like before anymore. So the key thing is that we need to make sure the cells is always in the exponential phase as much as we can, and give them plenty of nutrients and growth factors so that they can proliferate. And decline phase is when if the catalytic medium is not replaced accordingly, or and the cell number is not reduced, so the cells will start losing viability, will start dying, and the number will decrease. So this is another key thing we need to pay attention to, which is maintaining optimal temperature for cell growth. So most of the eukaryotic and animal cell lines require about 37 degrees Celsius for optimal growth. The insect and amphibian cells may require lower temperatures, for instance about 38 degrees Celsius. And some of the animal cell lines also they have temperature sensitive, and they could change their phenotype depending on the temperature. Most of the cancer cells can withstand the drop in temperature, and they can survive for a couple of days at 40 degrees Celsius. However, most of the cancer cells are very sensitive to heat. So if we heat them 2 degrees above the optimal temperature, just within a few hours, they can start dying. So we need to make sure that the temperature is optimal for cell growth. Okay, another one is